Hello friends, welcome back to another video. And in the previous video we discussed about the overview of glycolysis and also the whole cellular respiration. In this video we will discuss the events before occurring glycolysis. It means that we need glucose to be entered into the cell. Right now we discussed that how glucose will get into the cell and the process of glycolysis will be started. When we eat food, it goes into the stomach in GIT, and in here it's catalyzed into monosaccharides, especially glucose, fructose, galactose, and from here, from the GIT, it enters portal veins and go to the liver, and in the liver it means it modulates here, and after that it goes to the circulation, and from circulation freely it goes to interstitial fluids, and from here glucose cannot enter into the cell. It needs some special transporters to get into the tissue cells. So in here we discuss a little about the hepatocytes, what they will do with the glucose which is coming here. So hepatocytes has two main functions. One, balance glucose. It means when we eat food, glucose level will be very high, and right in here if it enters circulation it is dangerous, and we need glucose to be balanced in here, and hepatocytes function as a balancer for glucose and take much glucose inside and prevent from increase amount of glucose into the circulation. And the other function of the hepatocytes is to convert fructose, galactose, and other monosaccharides into the glucose in order to be usable in the body. So right now we discuss about the mechanisms by which the glucose enters into the tissue cells. Here we discuss about two mechanisms in which the glucose will enter to the tissue cell. First is facilitated diffusion, and the second one is glucose sodium cotransporter. So we discuss in here about facilitated diffusion. And facilitated diffusion, as its name declares, facilitated, it means transporters needed, and diffusion, it means from high concentration to the low concentration movement. And facilitated diffusion in here means that to diffuse by glucose transporters, to move by glucose transporters, or GLUT. GLU is taken from glucose and T from transporters. About 14 GLUTs are known, and most important are GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, GLUT7. We'll discuss about these GLUTs. GLUT1 and GLUT3, especially they are present in CNS and RBC, and their function is basal uptake of glucose. It means that on those situations that there is least amount of glucose in the circulation, they function to take the glucose into these important organs in the body. And these GLUTs, they have high affinity for binding to the glucose. The other one, GLUT2, it means, as its number declares, GLUT2 means two-way transport, especially it's present in the liver and beta cells of pancreas. In this GLUT, glucose can enter freely into the cell and outside the cell. Other one, GLUT4 is especially present in muscle and adipose tissue. This is very important to know that GLUT4 is insulin dependent. The other one is GLUT5. It's present mainly in testes and the GIT, and it's mainly fructose transporter. GLUT7, we discussed about GLUT7. GLUT7 is present in endoplasmic reticulum. It has a role in gluconeogenesis. So right now we discuss about the second mechanism of glucose transport into the cell, glucose sodium cotransport. So we consider here nephron in here, proximal convoluted tubule. In here it has sodium potassium pumps in basolateral side. It means it pumps sodium outside and potassium inside the cell, and the glucose sodium cotransport is present on the luminal side of this cell. And as the sodium potassium pump works, a lot of sodiums leave the cell, and amount of sodium inside the cell decreases, and the sodium glucose co-transporter in here function and cause that sodium come into the cell, and along with sodium glucose, which is coming from the circulation into Bowman's capsule, and from here along with sodium enters the cell through co-transporter. And glucose, after entering into the cell, it has special transporters in basolateral membrane, and from here it enters into the other tissue. So this process in here, sodium glucose co-transporter, is the result of active transport of sodium potassium ATPase. So guys, we end our video here, and we will continue the rest of the topic in another video. Please like, comment, share our video, and subscribe our channel. Thank you.